Hello, this is lecture number 16 that we're on now. We're going to be talking about hormones and neurotransmitters. And now that you have this background in terms of the synapse and action potentials, now we start getting into this area of research, which has become so important in the field of behavioral neuroscience, because if you talked with many neuroscience researchers now, they will tell you that this is where things are at. It's in terms of the biochemistry of these substances and what they're doing in the brain, and these gaps in these synapses, and the work designed to explore the relationship between this biochemistry and behavior is some of the most important work that's going on in the field, and we'll certainly get into this more and more when we begin to take a look at different behaviors and examine, the latest research that has been done looking at our brain biochemistry, namely these hormones and neurotransmitters and how they're involved in behavior. I start this out by encouraging you to think of your brain as being a factory, a chemical factory. I think that you need to have that firmly in your mind, that your brain is constantly in a production mode. And we've now been able to identify about 60 different distinct neurotransmitters in the brain, and we're learning more and more about them every single day. Again, we're going to take a look at what's happening in terms of the production of these neurotransmitters in the brain, help you to understand a little bit about the process that is involved in the manufacture of these neurotransmitters and the impact that they have on various parts of the cell. So neurotransmitters, again, these chemical events at the synapse. We'll begin that, and I have this little cartoon here that says, if I'm moody, blame my neurotransmitters, because neurotransmitters, whether we're talking about serotonin, norepinephrine, or dopamine, are very much involved in personality and mood changes. We're talking about the transmission of these neurotransmitters that are occurring at the synapse. Again, keep in mind your brain is like a giant chemical factory, constantly in a production mode. These chemicals, these neurotransmitters, travel across the synapse, and they enable this communication to take place between neurons. So again, it's important that we learn more about them. This is an important figure. It is one that I think we'll be referring to often. I want to make sure that you understand what the major events are that are involved in transmission that takes place at that gap, at that synapse. So let's take a look at this figure, and I'll point out certain parts of it. It'd be well worth your while to review it as we go along. Take a look at, here's our cell body that we see right here. Here's our axon that we see here. Here's the terminal ending or terminal button area right here. And if we blow up this area right here, it looks like what you see right here. Okay, so this is what we call the presynaptic area of the neuron, and you can see here this support cell that's in the green. This is our glial cells, which we've talked about in earlier lectures, that are so important for supporting the functioning of neurons. So one of the things that we know about these neurotransmitters is that some are actually manufactured here in the cell body. They typically are small molecule neurotransmitters, and what happens is they're manufactured here, and then they are pushed down the axon again, by way of these constant action potentials that are occurring. And they are in these little, what I call packages. They're referred to as vesicles, as you see here. Those vesicles, again, are transported with neurotransmitters inside of them down the length of the axon to this presynaptic area. And again, this right here, here's a blow up of it right here. So we can see then that these relatively large peptide neurotransmitters are in these packages, and they are transported down the length of the axon to the presynaptic area. And one of the things that happens is that that action potential is going to cause a lot of calcium to enter those packages and to release the neurotransmitters out into this gap, into this what's called the synaptic cleft. Smaller neurotransmitters like acetylcholine, for example, are actually synthesized in this area right here. So you have some that are being synthesized in the cell body, and you have others that are actually being synthesized in this presynaptic area that you see right here. So once those neurotransmitters, and those are in the red that you see here, are released, they're going to be taken up. They're going to bind with very specific receptors that are all along this postsynaptic area. And what we're also going to see is a separation that occurs from those receptors after activation has occurred, and they can actually come back in a process that's called reuptake, and they can influence the functioning of this presynaptic area in terms of the future production and release of neurotransmitters. One of the things that we know is that reuptake that occurs is very much related to these very specialized transporter proteins that bring that neurotransmitter back to this presynaptic area. We also have what is referred to as negative feedback. These are sites also that are all along this presynaptic area, and they respond to retrograde transmitters. And indeed, that too can modify what happens to the cell. We have then this very important process that's taking place in terms of the release of these chemicals, out into the synapse. And again, this is transmission that is occurring at the synapse. We've learned an awful lot about it in terms of what is happening biochemically, what is happening in terms of once those receptor sites in the postsynaptic area are activated, they can cause changes in this associated neuron here in terms of either increasing or decreasing action potentials. So again, this is very important to understand this basic process that is occurring here. The postsynaptic area that you see here, here's our receptors for these neurotransmitters that are being released by the presynaptic area. Once those neurotransmitters are released, they can occupy these receptors. It's almost like a lock and key arrangement. There are certain receptors that are responsive only to certain types of neurotransmitters. So this is, again, in the postsynaptic area. This is what we're talking about here. And we can either have excitation or inhibition as a consequence of those receptor sites being occupied. Here's, some major categories of neurotransmitters. Amino acids, peptides, acetylcholine, monoamines, purines, gases. These are the major categories of neurotransmitters. And remember, we have somewhere in the neighborhood of about 70 or 80 of them that have been identified. But here's some of the principal ones that you see here. Adrenaline, very much involved in stress responses. We call it the fight or flight neurotransmitter. Noradrenaline, very much involved in concentration. When you're highly focused on something, really concentrating on something. Dopamine involved in a lot of different behavioral responses, but namely in terms of mediating pleasure. So we call it the pleasure neurotransmitter. Serotonin, very much involved in mood and emotion. We call it the mood transmitter. GABA has a calming effect in the brain. We call it the calming neurotransmitter. Acetylcholine, very much involved in learning. We'll be taking a look at some interesting work when we get to the area of learning and memory that has to do with acetylcholine. Glutamate is also a very important neurotransmitter that's involved in processing of memories. And endorphins involved in feelings of euphoria. Indeed, these are, some of the major neurotransmitters that have been identified. I think principally the ones that we know the most about. So the synthesis of these neurotransmitters, how does this all occur? How do they form? Well, they really form as a consequence of your diet. And again, I have a few examples here that we can take a look at. Catecholamines are synthesized from the protein that's called phenylalanine. We get that from our diet a lot in dairy products, for example. Tyrosine. Phenylalanine converted to tyrosine, then ultimately to dopa, and to dopamine and norepinephrine and epinephrine. We also have acetylcholine, which comes from the acetyl coenzyme A that occurs from metabolism of various foods that we eat. It's converted to choline and ultimately to acetylcholine. Tryptophan, a lot of different sources for tryptophan, red meat in particular, poultry, where it's ultimately converted to 5-hydroxytryptophan and then ultimately to serotonin. So again, synthesis of neurotransmitters principally coming from those important dietary sources. Some facts about neurotransmitters in terms of their size and function. We took a look at this already a little bit, and we examined how these smaller neurotransmitters are principally formed in those axon endings. Acetylcholine, a good example. Larger neurotransmitters, they are produced in the cell body and again transported down by way of those vesicles down the length of the axon. These small molecule neurotransmitters typically act fast. But for a relatively short period of time. Neuropeptides, on the other hand, these large molecule neurotransmitters that we see being transported from the cell body, again, they're large and they're relatively slow in terms of their action, but they last a lot longer than do the smaller neurotransmitters. So some other facts about neurotransmitter size and function. Typically, transmission occurs very, very rapidly, across the synapse. Usually, in the neighborhood of about 10 microseconds. I mean, that's a very, very short period of time. So you're getting release, it's going out into the gap, and then it's being taken up by that postsynaptic area or by the presynaptic area. What we also know is that most neurons release a number of different neurotransmitters, 2 or more. Some cells, some neurons that release a dozen or so neurotransmitters. What we also know is that a neuron can respond to a lot of different types of neurotransmitters, many more than it can release, for example. So again, some other basic facts, about neurotransmitters. Reuptake and inactivation, we talked a little bit about that, but again, just to give you a little bit of a reminder here in terms of reuptake, that occurs in this presynaptic area that you see right here, in which it's taking up neurotransmitter molecules, it's taking them up intact, and it's reusing them, okay, for other purposes. They're being recycled. And again, they can be broken down by various enzymes within the cell. They can impact the future release of neurotransmitters from that presynaptic area. They have a lot of different functions that can occur. Transporters are very specialized proteins that are promoting this reuptake process. For example, serotonin, when it's taken back up into that presynaptic terminal area, serotonin attaches to these membrane proteins, and that's really going to promote that reuptake process. So when we talk about reuptake, again, here's a very simple diagram here to help you to understand this. When neurotransmitters are released out into the gap that you see here, they occupy those receptors in the postsynaptic area. They bind to them, and it's going to cause either excitation or inhibition of this postsynaptic area. But what we also have, of course, is this whole process that we call reuptake, where the molecule is coming back into the presynaptic terminal button area, and it's influencing various biochemical processes, a number of which are involved in the future release of that particular neurotransmitter. So, examples of inactivation and reuptake. Certainly acetylcholine is broken down by acetylcholinesterase. That occurs out in that gap, in that synaptic gap. It's broken down into acetate and the choline. Serotonin and catecholamine molecules are also converted into inactive chemicals, and MAO, the enzymes, COMT and MAO, convert that catecholamine transmitter into inactive chemicals. Inactivation of neurotransmitter, really there are three different things that I want you to think about in terms of this inactivation. First of all, neurotransmitters can be returned, as they are right here, into those axon terminals. They're going to be, they can be reused. They can be transported to glial cells, as you see right here. That's another thing that can happen to them. Enzymes can inactivate. And here's this inactivation that is occurring right here. They can inactivate neurotransmitters out into this gap. And neurotransmitters can diffuse into other areas, outside the synapse, outside the synaptic cleft, out into these blood vessels, for example. So let's talk a little bit now about hormones. Again, another very important chemical within the brain. We know that they're secreted by a number of different glands within our body. I'll have a picture of this in just a few moments. But they're transported by the blood, and they can have effects on many, many different target areas within the body, where they can alter the activity, the biology, the biochemistry of many, many different parts of our body. These endocrine glands, we'll discuss them and identify them in just a few moments. They're responsible for the production of those hormones. And again, the changes that they can produce, the changes that they can trigger, can be very long-lasting changes, and it can occur in many different parts of our body, as you will see as we go along. So here's a partial list of hormone-releasing glands. Let me just, talk about them briefly here. The hypothalamus, and again, I'll have some pictures of this in just a few moments, but they secrete a variety of different what we call releasing hormones that are impacting the pituitary gland. They also have behavioral effects that we're going to be taking a look at as we go along. The anterior pituitary and the posterior pituitary both are attached to the hypothalamus. The anterior pituitary has 6 primary hormones that it secretes. They're identified here. You know, I'm not going to spend the time to name them, but they do have very distinct effects in terms of our biology and our behavior. Just to give you an example, the hormone prolactin, for example, is very much involved in the production of milk in the mammary glands of mammalian females. But we also know that prolactin is involved in various aspects of parental behavior, maternal behavior. So it has biological effects and behavioral effects, and the same is true for these other hormones. The posterior pituitary gland, 2 principal hormones that are secreted, oxytocin and vasopressin, have, you know, just a whole array of different biological and behavioral effects. Let me just talk briefly about oxytocin because it will be coming up in other lectures, namely in the area of the study of parental behavior. But oxytocin is involved in controlling uterine contractions in the mammalian female. It's also involved in terms of the release or what we call the ejection of milk from the mammary glands. It's involved in parental behavior. It's involved in attachment and sexual pleasure. So a lot of different functions. And again, oxytocin, vasopressin involved in many different functions within the mammal. Again, we'll be talking about these things as we go along. Another hormone, melatonin, secreted by the pineal gland. It's involved in terms of the regulation of sleep, our sleep-wake cycle. The thyroid and thyroxine, for example, is involved in our metabolism. It's involved in how we mature, how we grow. The parathyroid hormones are involved in terms of regulation of calcium and potassium in the blood. The adrenal cortex, 2 hormones here, very much involved in terms of regulation of blood sugar, regulation of metabolism, proteins and fats. The adrenal medulla, epinephrine and norepinephrine, again, very important effects upon our sympathetic nervous system. The pancreas, insulin and glucagon, very much involved in the regulation of hunger and the storage of fat. Glucagon involved in the conversion of that fat into blood glucose. The ovary, the principal hormone secreting gland in the female. Estrogen and progesterones are secreted by the ovary, very much involved in female sexual characteristics and regulation of the female reproductive cycle. Progesterone very much involved in terms of maintaining pregnancy. The testis, and in the case of the mammalian male, we call them androgens. Many use the term testosterone, but has a whole array of different functions in terms of male sexual characteristics, both physical characteristics and behavioral characteristics. The liver and somatometins, that stimulates growth. The kidney and the secretion of renin, which is very much involved in regulation of blood pressure. The thymus supports immune responses, thymosin. And this last one, leptin, which is secreted from fat cells. It's very much involved in terms of appetite. We'll learn a lot about it as we go along in other areas of the course. So again, here's our endocrine secreting glands that you see here. You know, the testes, the ovaries in the case of the female, the pancreas that we see here, the kidney right here, the adrenal medulla that we see here, which is on top of the kidney, the liver that we see here, the thymus, the thyroid gland, parathyroid gland, and again, hypothalamus, pineal gland, and the pituitary gland. The hormones that are secreted from our endocrine glands are either protein hormones or peptide hormones, and they consist of these amino acids, and those amino acids attach to very specific receptors. We call them membrane receptors, and they activate something that's called messenger systems, second messenger systems, and that is all occurring inside the cell. And a very important messenger system is what we call cyclic AMP. Hormones secreted by the brain can control the release of other hormones, and I'll talk a little bit about that in our next slide. Here's our pituitary gland that you see right here in the red, and if we were to enlarge that, it would look like this. Here's the two parts of our pituitary gland, the anterior lobe and the posterior lobe of the pituitary gland. Here's our hypothalamus, here's the third ventricle. So this part of the pituitary gland, the posterior part, is actually an extension of the hypothalamus. And again, we have this membrane that covers this area. This is all at the base of the brain. This is bone that you see here, which is at the base of the cranial cavity. So the pituitary gland then has two distinct parts to it, anterior and posterior. Anterior pituitary gland is composed principally of glandular tissue, and it actually synthesizes 6 different hormones from this area right here. And the posterior pituitary gland that you see right here is principally neural tissue, and again, it's considered to be an extension of the hypothalamus. So take a look at another blow-up of this. Here's our pituitary gland. Here's what we call the pituitary stalk, and here's the hypothalamus up here. The hypothalamus synthesizes 2 hormones, oxytocin and vasopressin. And that oxytocin and vasopressin migrates down these axons to the posterior pituitary gland that you see right here. The hypothalamus secretes releasing hormones, which flow down through the blood system and stimulate the anterior pituitary gland to secrete 6 primary hormones: growth hormone, ACTH, which is called adrenocorticotropic hormone, TSH, thyroid stimulating hormone, FSH, and LH, and prolactin. So you have a very specific releaser hormone which finds its way down through our through the blood to the anterior part of the pituitary gland. And for example, what's called a CRF, corticotropin releasing factor, CRF, is manufactured up here, works its way down, and then stimulates the secretion of ACTH. We also have thyroid stimulating hormone releasing factor, manufactured up here, works its way down through the blood system and stimulates the production of TSH, thyroid stimulating hormone. So there's a releasing hormone then for each one of these 6 hormones, which works its way down from the blood system. It's manufactured here, works its way down, and then is involved in the stimulation of these hormones that you see here. So let's look at our last slide here, talk a little bit about hormones and feedback systems. The hypothalamus really is maintaining a constant circulating level of hormones. Remember I said the brain is kind of in a constant mode of production. Well, that's certainly what's happening in terms of the hypothalamus and these hormones, these releasing hormones, for example. So take a look at this. We have the hypothalamus that is causing the secretion of or promoting the secretion of TSH, releasing hormone. That's going to act on the anterior pituitary gland to promote the secretion of TSH. And that's going to stimulate the thyroid gland to secrete thyroxine. That gets out and has excitatory effects in many different areas of our brain, as well as in other parts outside the brain. And it also can feed back in an inhibitory way to suppress the release of more TSH. So this is a negative feedback system, and indeed is a very important one in terms of helping to regulate this steady state of secretion of this hormone from the hypothalamus. So in our next lectures, we're going to be taking a look at various drug effects and how they impact these neurotransmitter and hormone systems that we've just described.